Hello, College Biology. I'm going to try to make this video to last at 5 or 6 minutes, but because there are some complicated things going on and the phases of mitosis are kind of long, I'm going to have to talk pretty quickly. This is an experiment. We'll see how it goes. It's not really an experiment. It's a test, because if it were an experiment, I would be controlling multiple variables, and I'm not doing that. And I'd have multiple trials and all of these things, but that's not the case. So anyway, this is a test, and here we go. Let's find out. In the corner, mitosis. Mitosis is one type of cell division. Mitosis has some specific results and responsibilities. It results in two daughter cells that are each identical to the parent cell. So we want to do mitosis, and we don't want variation. We want things to be the same, such as when eye cells want to make more eye cells so that the organism can still see, or stomach cells want to make stomach cells, or heart cells want to make heart cells, or brain cells want to make brain cells, and skin cells want to make skin cells. So that's the deal about mitosis. Mitosis is responsible for building and repairing bodies. So little babies, they do more bodybuilding than I do because they're growing their bodies. So they have to do more mitosis so they can get more cells so that they can have a bigger body. You, high schoolers and college students, you guys are doing a lot of growth too. Even though your body may not be totally still growing, your brains don't finish developing until you're 25. So you're still doing mitosis to make new brain structures that I have already made. I am not doing that. I'm primarily doing repair. So when I get scratched or bruised or something, then I replace those cells via mitosis. So maintaining the body, because I'm losing cells all the time. So that's what mitosis is about. Now, let's look at the first phase of mitosis. This is actually not the first phase of mitosis. Interphase precedes mitosis. It's actually sort of the functional phase of the cell. This is when the cell is able to do its job, which it makes the proteins it needs to function in whatever type of cell it is. It's also the growth phase of the cell where the cells, take in material and grow and get fat. Now, toward the end of interphase, the chromosomes are duplicated, forming sister chromatids. Sister chromatids are two duplicated chromosomes that are connected to each other. If you ever see that X shape with the connection of that's actually two different there are two chromosomes that are identical to each other that have been copied, but they're not one chromosome. Chromosomes don't have an X shape. That's not true. Two chromosomes can be connected, which kind of makes them look like they have an X shape, but those are sister chromatids. Those are not actually single chromosomes, so they're one chromosome that's been replicated. So all of this is in preparation for mitosis. So let's take a look at what that looks like in picture form. That would be over here. This is our interphase cell. It's a cell. It's got its nucleus. It's got some centrosomes that are ready to rock and roll. But more importantly, it's got its DNA in the nucleus, and that DNA is unwound. It's uncoiled. If the cell's going to do its job, which happens in the functional phase of interphase, they've got to get to the genes that are on the DNA, so the DNA has to be uncoiled. So that's totally normal. So this is interphase, and this is the cell is going to be copying this DNA. Let's go to the next phase, which is going to be prophase, which kind of makes sense because like prologue, it's prefix, it means early. So this is actually the first phase of true mitosis. We're getting into our cell division right now. Now, these events, these don't occur step by step. They kind of all happen at the same time. Some may start before the other, but they're all kind of going on during prophase. First, the sister chromatids condense. This is when they're in their long stringy phase and they start to actually coil down into the visible X shapes that we can see under microscopes. The nucleus dissolves. During mitosis, we're going to be packing and moving around chromosomes. We can't do that if they're shut down in the nucleus. So we've got to take enzymes and proteins to help break up that nucleus so that we can move our chromosomes around. And then the centrosomes form spindles and push themselves to opposite ends of the cell. So it's kind of like the centrosomes start growing these spindles, and the spindles bump into each other and start pushing they keep growing. They start pushing centrosomes away from each other. Now, I can only go this far, but if I could keep making fingers that grow really farther, then they would keep pushing each other and I'd go farther. And that's what's happening to the centrosomes. And that's sort of prophase. So let's take a look at the picture of that. We've got our cell here. We've got our centrosomes forming spindles that are beginning to push apart. Our nucleus is beginning to dissolve, and our chromosomes are beginning to condense. Now, if we go over here, this is actually farther along in prophase. We've got our centrosomes that have pushed themselves to the opposite ends. Our nucleus is basically gone, and our chromosomes have tightly condensed down. Now, they're still kind of scattered about, which brings us to our next phase, which is metaphase. In metaphase, the spindles attach to the centromeres of sister chromatids. Whoa! Okay, the spindles attach to the centromeres of sister chromatids. The centromere is that attachment point, so the sister chromatids are attached to each other, and the centromeres also attach to that. Now, the centrosomes then use the spindles to alternate pulling So they do this sort of like back and forth, taking turns pulling on the chromosomes until they can get the chromosomes in the middle of the cell where they need to be. So those sister chromatids get all lined up. That is what this looks like down here, right here. This is our metaphase. So now we have spindles that have attached to the centromere of our sister chromatids, and they've done some tug of war, some back and forth to get them lined up in the middle, which means we are now ready for anaphase. In anaphase, the centromeres reel in, they digest the spindles that are attached to the sister chromatids. Well, if they're both attached to sister chromatids and they get pulled apart, well, they kind of break apart the sister chromatids. So now those duplicated chromosomes are each going in different directions. They're being pulled to opposite ends of the cells. Meanwhile, the spindles that were the pushing spindles, they're still growing, they're still pushing, and the centrosomes are being pushed even farther apart from each other. So we've got the chromosomes being pulled toward the ends and the centrosomes being pushed even farther apart from each other. That's all going on in anaphase. And what does that look like? Well, we can see the pushy spindles here, and then we can see the other spindles beginning to reel in the chromosomes there, which brings us to the last phase, which is telophase. In telophase, the spindles are digested, 2 nuclei reform, the rest of the organelles in the cell kind of get divided to either side, and then the cell undergoes cytokinesis, which is the process of cutting the cell, where the membrane pinches to form 2 daughter cells that each have a nucleus with all of those copied chromosomes in them. And so what does that look like? That looks like this. We've got our nucleus reforming. We've got our chromosomes. They're going to uncondense because if they're going to need to be useful in the next cell. And we have cytokinesis occurring and the separation of our organelles to give us 2 daughter cells. I'm going to real quick go back here and say that I'm going to add it because it needs to be added. Chromosomes uncoil. And that's necessary. They're chromosomes. Chromosomes uncoil. And my goal to be done with this video in 5 to 6 minutes has failed. It is a 7-minute video. Tough. Struggle well.